Okay, this is my case study for penicillin and bacterial cell wall permeability presentation. So to begin, what is penicillin? So penicillin is a class of antibiotic medications that are used specifically to treat bacterial infections, and they do so by attaching and damaging the cell walls of bacteria. Some of the infections that penicillin treats include strep throat, ear infections, staph infections, salmonella, syphilis, Lyme disease, and more. There are three different types of penicillin. The first is natural penicillins, such as penicillin G and penicillin V. These are not made by people and are isolated, then purified amounts of penicillin that are found in the world to be made into medications. The second type is called semi-synthetic penicillins, and these include penicillase-resistant penicillins, aminopenicillins, and extended spectrum penicillins. And they are the alterations of penicillin's natural form to make it into a more effective antibiotic. And the last is combination medications. These are doses of penicillin combined with other medications to create one single effective drug. So, penicillin has a very important function in killing bacterial infections, and this is through the interruption of bacterial cell synthesis, specifically through the inhibition of the enzyme penicillin binding protein, also known as PBP. The structure of bacteria includes cells being enclosed within a cell wall, which is formed through many peptidoglycan chains being cross-linked through the action of PBP. The enzyme DD transpeptidase is used to create a net-like composition that helps to support and protect the cell. Bacteria cell permeability is very specific, with outer membranes of gram-negative bacterial cells being the main line of defense. They work to filter what can exit and come into the cell. And because of this, it makes the cell walls a main target of the antibiotics, but also helps to protect them. In contrast, gram-positive bacterial cells are often less resistant to penicillin, and this is because they have thicker layers of peptidoglycan that are more heavily targeted. Penicillin is able to kill bacteria through the binding of beta-lactam ring of DD transpeptidase, which is then able to directly inhibit cross-linking activity, which then leads to the prevention of new cell walls being able to be created. So, without having a cell wall, bacterial cells are incredibly weak and sensitive to molecular pressures and any interactions with water, and this will then lead to a very quick cell death occurring. It is able to avoid interacting with human cells in the body because our cells do not have cell walls, so the penicillin treatment will only target these bacterial cells. So, how bacteria evolved to resist against penicillin? So, bacteria are able to quickly reproduce and able to avoid genetic mutations even when in the presence of pressures around them, such as antibiotics being introduced to the environment. And this is because they do not evolve to rid of their cell walls. That being said, their cell walls begin to grow resistant to these antibiotics because of such, and this could be through the repeated or the improper use of antibiotics, and they will begin to show changes of genetic mutations, and these will arise in resistant strains. Ultimately, this leads to the necessity for stronger antibiotics to be used to be able to treat and get rid of the bacteria. So, in conclusion, penicillin is a very effective drug in its ability to target bacterial cell walls. They're able to avoid the harm of other healthy human cells. Penicillin is able to inhibit the synthesis of cell walls in bacteria because of PBP enzymes binding to them. While bacteria does have the ability to mutate in strains to create resistance to these antibiotics, it is not always effective and can be limited through ensuring that these drugs are not overused or improperly used. And to the right, you could see my chimera structure of penicillin. And here are my citations. Thank you.